The part 3 notes on plants is going to be on water transport through xylem tissue. So remember from BioH, we talked about ways that molecules can be transported across a cell membrane. And we said that these processes can occur either passively or actively. We said that in passive transport, it involves moving molecules from an area of high concentration to an area of low concentration. Since we are moving from high to low concentration, no energy is needed. In BioH, I said this is like rolling down a hill or sledding down a hill. It does not take very much effort or energy out of you to go down the hill. Once you start going, you just keep on going. So we talked about 3 types of passive transport. The first type we said was called diffusion or simple diffusion, same thing. In diffusion, molecules are moving straight through a plasma membrane or straight through a lipid bilayer from high to low concentration. So really good examples of this would include in the root cells, oxygen diffuses into the root cells. It's going to use this oxygen for cellular respiration, and then CO2 will diffuse out of the root cells. In facilitated diffusion, molecules are still being moved from high concentration to low concentration, but they can't go straight through the lipid bilayer. This is what makes it different from simple diffusion. In facilitated diffusion, it has to go through a protein channel. This is because the molecule is either too big and or too charged to go through the lipid bilayer. So a protein channel facilitates the movement of this molecule from high concentration to low concentration. To facilitate means to help. So the protein channel is the helper. So we're still moving molecules from high concentration to low concentration, but this time we're going through a highly specific protein channel. That's facilitated diffusion, right? Whereas this one is more like simple diffusion. Osmosis is a special type of diffusion. It is specifically the diffusion of water from an area of where there's more water available to the area of where there's less water available. So water this time is moving from high to low. And a good example of this is water has to move into the root cells by osmosis because we need water to fully access the plant, right? To get up to the leaves, for example. And so how we get it, though, is through osmosis into the root cells. This means there would be more water available in the soil and less water available in the root cells, causing water to move into the root cells by osmosis. So in osmosis, in BioH, we also used these vocabulary terms, hypertonic, hypotonic, isotonic, right? We said whichever side of the solution has more solute dissolved into it, that side of the solution is hypertonic, and the other side is hypotonic relative to the hypertonic side, right? So hypo means less or under, and hyper means more or elevated. So when we look at this U-tube example, which is separated by a semi-permeable membrane, this side has less solute concentration dissolved within the water. So this is the hypotonic side. This side has more solute dissolved within it, so a higher concentration of solute in the same amount of water. And so this side is hypertonic. Water always moves towards the hypertonic side. So from hypo to hyper, because it's trying to dilute this side of the membrane. So water moves to the right by osmosis, and as a result, you see the water level rise. So this is because there's more available water to move from the hypotonic side, and water moves from high availability of water to low availability of water. And this is shown more specifically in this image. So you can see the purple stuff is the solute. The solute cannot cross the membrane. The membrane is not permeable to it, but water can cross the membrane. So there's less solute on this side, more solute on this side. Water is taken up by the solute, some of it. It's bound to the solute. It's attracted to the solute because it's trying to dilute or dissolve what's happening with the solute within the solution itself. So all of these water molecules are free and available to move, whereas these ones are not. So there's more available water for movement on the hypotonic side of the membrane because less is taken up by the solute. Therefore, water moves from high water availability to low water availability. Net flow of water is to the right in this situation. Net flow means that there is an overall higher flow in one direction. And so we also use the terms isotonic, right? So if the two solutions are not hyper and hypotonic, if they are equal to each other in concentration, then they would be isotonic to each other. Like an isosceles triangle has two equal sides. The two sides of the membrane are equal in concentration. So at that point, there'd be no net movement of water. Water would move back and forth at equal rates. Moving on to active transport. So we said that molecules can move actively across a membrane as well. In the case of active transport, we're moving molecules against their concentration gradient. So molecules are moving from low concentration to a higher concentration. And so this is typically done by using transport proteins that require energy. So starting off with protein pumps. Protein pumps typically use ATP energy, not always, but do typically use ATP energy. There are other types of energy that can be used, but they typically use ATP energy to move molecules from an area of low concentration to high concentration. This one specifically shows a proton pump because we're pumping protons from low to high concentration, or hydrogen ions from low to high concentration. We're pumping them from inside the cell to outside the cell. This generates what's called membrane potential. Membrane potential, remember, from BioH is a difference in charge or voltage across the membrane. So by actively pumping out more positive ions than are found inside the cell, the inside of the cell maintains a negative interior relative to the positive exterior. That's important for plant cells. Okay, so we're going to come back to that, but we are generating what's called membrane potential and a gradient of hydrogen ions. So there's a difference in both charge across the membrane, the outside's more positive, as well as a difference in concentration. We are concentrating the hydrogen ions on this side of the membrane, and that is done through active transport. Another type of active transport involves co-transporters. So this typically involves two substances moving through the same protein and also involves chemiosmosis. So we will talk about this more on the next slide. So typically co-transport molecules, which here's the co-transporter in this example, often involves a coupled channel. So some other channel that's working in conjunction with it. So let me explain that. So here we have our hydrogen ions being pumped through this proton pump from low to high concentration. We spent energy to do that. And so that's just like what we talked about on the last slide. Like the last slide would have just been this molecule. But why that's important to the co-transporter is because now that we have this buildup of hydrogen ions out here and they are in high concentration, they are going to diffuse back down their concentration gradient by facilitated diffusion from high concentration to low concentration back into the cell. And we don't have to spend any more energy to do this because, again, they're going down their concentration gradient to come back in. But this second channel that we're using is a co-transporter. Specifically, it's transporting not only the hydrogen ions down their gradient, but it's also transporting sucrose molecules with it. So at the same time, through the co-transporter, hydrogen ions come in, and so does sucrose. The neat thing about this is sucrose is being moved against its concentration gradient from low concentration to high concentration. And we're not spending any additional energy to do that. We already spent the energy from this pump. right? So that's the chemiosmosis part. We spend energy, right? That's our exergonic reaction here. Energy was just spent. We built up this gradient because of that. But when they come back in, we don't have to spend any additional energy. And the gain inside the cell is we've just loaded a bunch of sucrose inside the plant cells. And so this is important when we talk about the transport of sucrose in the next set of notes. So this is how cells, plant cells, specifically are going to load a bunch of sugars into the sieve tube members so that we can transport them to other locations throughout the plant. Here's another example of a co-transporter, and it likely involved a proton pump above this as well. It's just not shown in this picture to generate this gradient over here. So the hydrogen ions are pumped out first, but then this co-transporter is going to bring nitrate back in with it as hydrogens are coming back down their concentration gradient. And the nitrate ions are being moved against their concentration gradient. So again, it's like these other molecules are kind of catching a free ride. They get to go against their concentration gradient without spending any additional energy. We already paid with the energy from the proton pump, for example. And so nitrate is just another example of how an ion can come through a co-transporter. And so this would be like nitrates in the soil, for example. But it could be other types of ions in the soil as well that we need to get into the root cells. Here's another picture showing the coupled channels and the loading of the sucrose into our sieve tube member or into a phloem cell. So same thing. We first pump it out. We paid for that. We get a buildup of hydrogen ions that then come back down their concentration gradient, and they bring sucrose with them through the co-transporter against its concentration gradient. Sucrose is catching a free ride here because we already paid with the proton pump. So we're constantly moving mineral ions into the root by active transport, and this allows for us to concentrate those ions inside the root cells at really, really high levels of concentration. The root cells themselves can be 100 times more concentrated in these ions than the soil itself. And again, this is due to active transport. We're actively transporting these ions into the cell. And it's highly specific how this is done. So each specific protein pump within the root cells is unique to what mineral it can transport in. So it's not like they're just going through a whole bunch of different protein channels. The nitrate can only come through a specific pump. Phosphate can only come through a specific pump. Potassium can only come in through a specific pump. So again, they're highly specific. This picture down here shows like a bunch of specific pumps. And they typically take ATP to get these into the root cells. Some ions move very slowly through the soil because they become very attracted to the soil, and then they kind of get stuck on the soil. They bind to the soil particles, and that makes it hard to transport these particular ions into the root cells. But there are ways that plants can overcome this. There are several ways. We will only talk about this one specific way right here, which is only specific to some types of plants. So some types of plants, like some trees and orchid flowers, for example, their roots actually have a mutualistic relationship with a fungus. And the fungi, they have kind of a root system. They're called hyphae. You can see that here. Like here's the fungus, here's the hyphae. They look similar to roots, but they are structurally different. And they associate, like grow into the roots of the plant system. And by doing this, they share things with each other. The fungus is sharing nutrients that the tree normally wouldn't have very good access to. And then the tree is feeding the fungus with carbohydrates that it's made in photosynthesis. So again, they are working together in a mutualistic relationship, benefiting from each other. And this picture kind of shows the same things. Here's the fungal hyphae. And here are like the root cells as well, and they associate together. This active uptake of ions into the root cells is important because the root cells then concentrate this in such high amounts inside the root cells, and this is what drives water into the cell, right? So wherever the solute is, water will follow. So the more steeper the concentration gradient is, like the more ions that we have concentrated into those root cells, the more water that will follow and go into the root cells as well. So it's very important for water uptake. And because we're constantly doing so much active transport in the root cells, these root cells are going to have lots of ATP energy. Specifically, these are going to be in the epidermal layer, because that's the first layer that would have the proteins to pump these molecules in. So in that layer, they are rich in mitochondria, and they need a constant supply of oxygen because we need to constantly be doing cellular respiration to make ATP to be able to pump these ions in such large amounts. But again, as the mineral ions are pumped in by active transport, water will follow by osmosis. What specifically, though, is going to cause the water to move? And this is going to be due to differences in water potential. This is where we kind of take it to the next level in regards to osmosis compared to BioH. We didn't talk about water potential in BioH, but what water potential is it allows for us to predict the direction that water will flow from. You must highlight, underline, circle the word from. Water potential predicts the direction that water will flow from. So it's going to move from this side of the membrane to the other side of the membrane. Whichever side has a higher water potential, water will move from that side. Water potential includes the combined effects of solute concentration, which we call osmotic potential, and pressure potential, which has to do with the differences in pressure in and out of the cell. And so it is a math equation. When we look here, you can see that we have our water potential is equal to the pressure potential plus the solute potential. This is very similar to a lab that we did in BioH, where we used dialysis tubing, and we put a solution inside the tubing, and then we put it in a beaker in a different solution. And so we were able to compare the concentration of solute both in and out of the fake cell. Our dialysis tubing was our fake animal cell, and predict the movement of water. In our lab, not only did we predict it, but we masked the bag of dialysis tubing both before and after like a 30-minute wait period, and then we used the differences in mass to determine if in fact we did predict the direction of water movement correctly. So in this example, you're going to want to pause the slide and predict which direction do you think water will move in or out of the cell. Hopefully you said that water will move out of the bag because there is a higher concentration of solute outside the bag and in the beaker. So water always moves from the hypotonic side inside the bag towards the hypertonic side, which is outside the bag. It wants to dilute that solution. So water is moving out of the bag. In animal cells, we did not have an issue of pressure potential because in animal cells, they don't have cell walls that are capable of pushing back. So in animal cells, their solute potential or osmotic potential is actually equal to their to their total water potential. Those two values are equal to each other because there is no pressure component. That's why we didn't really talk about this equation in BioH, and we saved it for IB because this is when we really get into the details of plants. Okay, so again, in animal cells, there's no pressure component. So their osmotic potential, which we did talk about, was really the same as their water potential. So again, here's our equation. Total water potential is equal to the osmotic potential plus the pressure potential. As the solute concentration increases in solution, the osmotic potential actually decreases. And so that means that value is going to become more negative. This is because we're always comparing to pure water or distilled water, which would have nothing in it. So pure water always has an osmotic potential of 0. So that means if we're always comparing to 0, then it would always just go down from there. Therefore, it becomes more negative. Pressure is usually opposite. As the pressure increases, the pressure potential becomes more positive. So you're going to see that number get bigger. But it's really important that you always use the total water potential when predicting the movement of water. So when we look at the U-tube on the right, which is like a plant cell, how a plant cell kind of operates, on the left side, we have our distilled water or pure water. So there's nothing dissolved in it. So we know that osmotic potential is 0. But in addition to that, notice that there's no pressure exerting any force on the left side of the U-tube. This is because it's considered to be an open container. So it's open at the top, no pressure. Anytime you see open container or it's in an open beaker, that means pressure is 0. So 0 pressure, 0 osmotic concentration, that means total water potential is 0. But when we look at the right side, you can see that we do have solute, and we're given that the osmotic potential is -1, so that's down here. And then we're also given that the pressure potential is +1. So we have pressure pushing back at the same amount of force that the solute is concentrated within that solution. So even though we do have a pressure component, because those two values are equal to each other, our total water potential is still 0. Therefore, there would be no net movement of water. Water would just move back and forth at equal rates because water always flows from a higher water potential to a lower total water potential. Water potential is the potential energy of water to move per unit of volume. To be able to quantify potential energy is very challenging. The absolute quantity of potential energy cannot actually be determined. Each value is really just relative to another value. That means we have to always have something to compare to. So in the case of water potential and the potential energy in regards to that, we're always comparing to pure water at standard atmospheric pressure at 20 degrees Celsius. And so if this is true, we have assigned that to have a water potential of 0. And so we'll always compare to a water potential of 0, but again, that's at normal pressure at a specific temperature with like nothing dissolved in it. And that's where that comes from. Like that's how we have something to compare to. The more pressure that's being exerted, the more potential energy that water will actually have. This would mean more water is likely to move from that solution due to the pressure. Okay, so more pressure means more potential energy, which increases the water potential. Water is more likely to move from that solution because of the pressure. But we also have to consider the solute concentration. The higher the solute concentration, the less potential energy that water has. That would mean that water is less likely to move from the side that has a higher solute concentration. So that means that if water is less likely to move from that side, there is a lesser water potential on that side because it has less potential energy. So water moves from high to low water potential because this minimizes its potential energy. Remember, that's like rolling down a hill. We want to spend less energy, right? So less potential energy to move water from high to low concentration. This slide shows 4 different U-tube circumstances, and they all kind of mean different things. So if we first look at A, U-tube A over here on the left, this one's like a mock animal cell because we don't have a pressure component. So on the left side, we have pure water. in an open container. So again, no pressure and no osmotic potential. Therefore, total water potential is 0 on the left. On the right, we're still in an open container, so we don't have pressure. Pressure potential is 0. Sorry about that. So on the right side of the YouTube right here, we don't have any pressure since it's an open container, but we do have solute. And so the solute potential is given to you. These numbers either have to be given to you or you have to have enough of the other numbers to calculate it. So if we do the math, our total water potential then, 0 minus 0.23, is going to give us a negative 0.23 for our total water potential. And so water always moves from the area of higher water potential, the bigger number, to the area of lower water potential. Therefore, 0 is a bigger number than negative 0.23. Water is going to overall move to the right. So again, this is like an animal cell because there's no cell walls pushing back, so no pressure component. The second image has to do with a mock plant cell because now we have that pressure pushing back. So on this side, again, it's an open container with nothing in it, no pressure, total water potential is 0. On this side, we do have stuff dissolved in it, so we have our solute potential, our osmotic potential, but we also have pressure pushing back with that positive force. So because they're equal to each other though, that still gives us a total water potential of 0. That means water moves back and forth at equal rates. So still no net movement of water in this image. These last two are not natural environments. These are more like no real cell is going to react this way, but they are more like potentially if you could apply more pressure or pressure in a different way, could you force more water to the other side? And so when we look at C, again, open container on this side, so 0 for total. And then here we have our solute. So again, osmotic potential is still negative 0.23. And our pressure is pushing down at a really, really like positive value, a higher value. So our pressure potential piece this time is high. And so this time we have a total water potential of 0.07. And so could we, if we continue to apply more pressure like this one, force water to move onto this side? Yes, we can. Pressure can force the water to that side. But a plant cell would never do this because that would mean the plant cell is literally pushing water out of its cell. So why would it want to do this? But this is actually the premise behind what's called a reverse osmosis water system. So if you've ever heard of that, or maybe you have one in your house, this is what's happening. You have your water with stuff dissolved in it that you don't want to drink, and you have your holding tank that's full of clean water that you're drinking, and they're using pressure to force more water into your holding tank to give you that drinkable water. And this one, again, no natural cell is going to react this way, but this is saying no pressure on this side. We still have our solute, but what if on this side we applied a negative pressure? So we're no longer in an open container. This time we're pulling. So if there was enough pull, that's called tension or negative pressure, could we potentially pull water up to this side of the membrane? And yes, we could. If this value was big enough, right? If we had enough tension, enough pull, like a suction or like a vacuum, right? We could pull the water to this side of the membrane. This slide is to remind you that we've been looking at these U-tubes, but it's really just a model for our cell, right? So here's our plant cell. You have the pressure it's exerting by having water build up inside the cell. That puts a lot of pressure on the cell walls, and then the cell wall is like pushing back. And so again, that pressure difference is what is really kind of keeping that water like sort of swollen inside the cell. And so this is kind of similar to like if you've ever pumped up a basketball or pumped up a bicycle tire with a hand pump. And at first it's like pretty easy, you're putting a bunch of air into it, but at some point it becomes very challenging to continue to push more air into that tire or into that ball. And that's because the tire or the ball is like pushing back with a force. And so that push back is what causes it to, not be able to really take in any more air. And so we're getting that pushback from the cell wall. Remember this lab from BioH, we did a lab on red onion cells where you first soaked the cell in distilled water, and we saw those like nice swollen vacuoles as water constantly went into the cell. And then we redid it with salt water, soaking it in salt water solution, and that caused water to leave the vacuole and then leave the cell. So you saw your vacuoles kind of like shriveling up as water was leaving the cell. At that time, we just used the terms hypertonic, hypotonic, and talked about the type of osmotic environment that we created for the cells. And that is all still true. But what we did not talk about is the total water potential. And that's really what you were changing, is the total water potential. So I want you to pause the notes and think about how were we changing the total water potential in these two environments? Like, where was water potential higher? Outside the cell and inside the cell. Do this for both environments. So hopefully you looked at this environment and you realized that since water is moving into the cell, we created a higher water potential outside the cell. Water is going to move from that side into the cell, right? So when we created that distilled water or hypotonic environment, what you were really doing was you were changing the water potential outside the cell. You are raising the total water potential, and that caused water to go into the cell. This creates what we call turgid cells, swollen, plump cells, due to the turgor pressure. When a bunch of water goes into the cell, that water puts pressure inside the cell. That's called turgor pressure, and then the cell walls push back. In this picture, hopefully you said the opposite was true, right? We had water leaving the cell. Water always goes from where there's a higher water potential to where there's lower. So you created a lower water potential outside the cell due to the solute. This caused water to come out of the cell. And so when that happens and the plant's losing a lot of water through osmosis, that's not good. That's called plasmolysis. If plasmolysis occurs, then what's happening is so much water has left the cell that it actually causes the plasma membrane or cell membrane to plasmolyze, which means to like pull away from the cell wall. And overall, you would see that kind of shrivel up. If this was your plant that this was happening to, you would see your plant wilt, and eventually you could see your plant die. And this is just another picture, showing you that. So we do have like the cell walls, pushing in this picture right here, but we would want lots of water inside to have that turgor pressure to build up, so that they kind of like push against each other and maintain a nice plump cell. Remember that the cells need that water for not only photosynthesis, but also water inside the vacuole provides strength and support for the plant. So again, it's really important that plants have a bunch of water inside their cells. And again, water doesn't just go into the cell and sit in the cytoplasm. Water specifically goes into the cell and then into the vacuole. So that is where the water will be stored, is inside the large central vacuole for plants. Another term for this large central vacuole would be a sap vacuole. A sap vacuole means that it's some kind of vacuole that's like filled with liquid, typically water. And the large central vacuole is a really good example of a sap vacuole. But there are other types of sap vacuoles also. And I'm saying sap, like sap. Okay, so how does water move short distance ways in a sort of lateral movement? And so again, lateral movement or short distance transport would mean we need to get water from the soil all the way through the root tissues and then into the vascular tissue, which is the stele, right? Because once it hits the stele, it's now ready for long distance transport to go up the cell, or sorry, up the plant, up the stem, so that it can be transported to all places around the organism. So again, let's review part of this short distance transport, and then I'm going to introduce something new right here. So minerals have to be actively moved into the root cells. and that allows for them to be highly concentrated. Once we highly concentrate those minerals in the root cells, water will follow by osmosis because the roots are hypertonic, right? So if the roots are hypertonic, we would have a lower water potential inside the root cells, higher water potential in the soil, and that's what forces the water to move into the root cells. But once water moves, into the plant itself through the roots, water has to be checked out by at least one root cell within the root tissue system before it can get to the stele. So by checked out, I mean that water has to be able to go through a plasma membrane of at least one cell. So at least one time, it must go through a plasma membrane because there are proteins in the plasma membrane that will regulate the movement of other substances into the cell, right? So we don't want certain things to come into the cell. We only want what the plant needs to come into the cell. And so this is really important because if we don't go through a membrane and have water get checked out at least one time and it enters the stele without getting checked out, well, the stele is like an elevator and it gives it full access to the entire plant. So that means water and anything transported in it or dissolved in it is going to go upwards in the plant. And so again, we only want those things that the plant needs or wants to make it to the stele. And so when we look at the way the root is organized in regards to tissues, here's our soil, here's our root hair, right? Our epidermis. Water's going to go through the epidermal layer, then through the cortex, and then the last layer of ground tissue right here is our endoderm, right? So this endodermal layer right here is the last chance water has to be checked out, because if it crosses the endoderm and into the pericycle, we're now into vascular tissue, and then it will get into the xylem tissue in the stele. Okay, so we're going to now talk about this like last chance to get checked out in the endoderm. So here shows you different routes that water can take. We're going from the soil into the epidermis first, through all this ground tissue, the root cortex, and eventually into the xylem. But there's multiple ways water can do this. Here's one way if you follow the blue line, right? So water comes in. You don't need to know this term, but water comes into the root cell. So now right here is the membrane. So it has gone through a plasma membrane and it's now into the root cell. So it got checked out. It only needs to get checked out one time. It can get checked out more than once, but it has to get checked out at least one time. So this water did get checked out. And so now it's in a cell and it can just move through plasmodesmata from cell to cell to cell, eventually crossing the endoderm and making its way into the stele. So that's one way, right? It gets checked out right away. Or water is actually so small that it can travel between the cell walls of plant cells. So here it is coming in. It's not in a cell, right? It's like in the cell walls. We call this the apoplast. The cell walls are the apoplast. So it's moving like between the cell walls of the different plant cells. If it continues on this pathway, so follow the red line, in between cell walls all the way through the cortex, then eventually it's going to make its way to our endodermis layer right here, which again is our last layer of ground tissue, and it will have not entered through a cell membrane yet. So we need it to enter a cell membrane before it gets into the stele. So it's gone this apoplastic route, and ultimately it's going to hit a barrier right here between the cell walls of the endodermis. You don't really need to know much about this barrier, but I am going to explain it to you because I think it helps understand the concept of water getting checked out here. And so this barrier right here is hydrophobic. And so once water gets to that barrier, it can't cross, right? Because water is so polar and this barrier is strongly nonpolar. It's hydrophobic. It repels the water. So it's like water hits it and then it gets stuck and it's going to be forced to go into an endodermal cell. Well, when it's forced to go into this endodermal cell, it crosses a membrane. So it got checked out. So now it's in the endoderm and it can go into the stele safely. And water can actually go back and forth between the cell walls and into the cells. So like if we follow this apoplastic route, you can see it came in through the apoplastic pathway, but then here it entered the cell. So it can cross the membrane here, right? It got checked out. So now it can continue to move this pathway. And you can see that it goes in and out, in and out, in and out. That's normal too. And it can get checked out more than once, but it has to get checked out at least one time. If water sneaks through the apoplast all the way to the endoderm, then it hits the barrier and gets forced into the endodermal cell, and that's where it gets checked out. So once it's checked out, it enters the xylem tissue, and it's ready for long distance transport. So again, all minerals that enter the stele have to pass through at least one selectively permeable membrane, at least one plasma membrane in a root cell. So this can be done at any point along water's travel. It can be through any cell in the root. It could be through the epidermal cell. It can be through any of the cells in the ground tissue. But the last chance for this to occur will be at that endoderm. So again, as it's coming through, if it has not yet entered a membrane, it's going to enter here because this hydrophobic barrier forces water into the endodermal cell where it gets checked out. Last chance to get checked out. Once it's checked out, it's now entered the stele in the xylem tissue, and then we're ready for long distance transport. So this is where I'm going to stop the notes, and the next recorded lecture will be the rest of part 3, which will be the long distance transport of water up the stem.